Today I'll be talking about protein digestion, which is actually fairly straightforward. So what happens when we eat protein? Well, in the mouth, obviously, we're going to moisten and break down that food and chew it. Anybody that has issues with saliva production may have difficulty with this and may, in fact, have something called dysphagia, which is difficulty swallowing food. And the stomach is where proteins are denatured. That starts with hydrochloric acid, and then digestion actually starts with pepsin. This is the 30,000-foot view, just a brief overview. Small intestine, we have trypsin and chymotrypsin coming from the pancreas to break down polypeptides. Remember, polypeptides are just chains of amino acids linked together by peptide bonds. They can't be absorbed intact, so they have to be broken down into shorter peptides, or actually individual amino acids. Where are we getting amino acids? Well, obviously from protein. Amino acids are the building blocks of protein. So there's exogenous sources, which are actually what we get from food, so dietary sources, what you would think of typically: meat, poultry, fish, eggs, legumes, nuts, seeds, some vegetables. Endogenous protein comes from sources within the body which are broken down or degraded. For example, we might swallow mucosal cells that slough off, the intestinal tract turns over, and that material is digested and actually a source of protein. So if we take a closer look at digestion and we look at the stomach, we know that gastrin is released by the pyloric, in the area of the pylorus in the stomach, into the bloodstream, and that's going to stimulate the release of hydrochloric acid. What does hydrochloric acid do? Well, we talked in the previous lecture about denaturing different structures of protein. So we have the quaternary, tertiary, and secondary structures, which are actually folded up proteins, and those have to be opened or denatured. Pepsinogen is an inactive zymogen, which must be made active, which is done with hydrochloric acid. So pepsinogen becomes pepsin, and then pepsin in turn hydrolyzes those peptide bonds which are linking the amino acids together, which we need to break so that we can digest the protein. In the small intestine, we have these partially digested proteins which are entering, and then secretin and cholecystokinin are released, as I mentioned, kind of in the 30,000-foot view, as I like to call it. Proenzymes and bicarbonate are released, and that neutralizes the acidic chyme in the stomach. And we have inactive enzymes, which are converted to active enzymes, and those tri and dipeptides are actually freed or broken so that we have, or the bonds are broken so that we have free amino acids. And there are a few other intestinal enzymes that play a role as well. In terms of absorption, the free amino acids are absorbed from the lumen of the small intestine, and there's two intestinal membranes that must be crossed in order for those amino acids to enter the blood and be transported to the various tissues in the body. The first one is the apical membrane, also known as the brush border, and the serosal membrane, also known as the basolateral membrane. In terms of intestinal cell absorption, so we're talking enterocytes, the amino acid, the free amino acids, cross that brush border membrane, and that usually happens in the duodenum or upper jejunum. Now, if you might recall in the Roux-en-Y gastric bypass procedure that we talked about, that first portion of the small intestine, or the duodenum, and actually the first proximal third of the jejunum are bypassed, and often people with who have had this particular type of weight loss surgery have an intolerance to meats, which I'm not sure at this time if they've actually definitively figured out why that happens. But again, interestingly enough, the absorption of these amino acids occurs in those two areas in the small intestine. So we are looking at potential deficiencies due to malabsorption. Carriers are required for that amino acid intestinal cell absorption. So we have passive diffusion, where there really isn't a carrier required. Active carriers are driven by ion gradients, and then the sodium pump is the third type of carrier that's required for absorption or utilized. In terms of extraintestinal cell absorption, so we're talking about absorption outside of the small intestine, we have amino acids entering the portal vein, and that's how they make their way to the hepatic tissue or the liver, and then they enter into the hepatocytes. They are transported, obviously, to other cells in the body. There's something called the gamma glutamate cycle, and I don't want to say it's controversial, but I'm not sure that it's widely accepted. It's been talked about since the 70s, but that's a type of transport that occurs in the renal tubules. It's theorized renal tubules, erythrocytes, and possibly neurons. So what happens after absorption? Well, we talked about the amino acids entering the portal vein and then traveling to the liver. The liver is going to keep some of those amino acids and, use it for its own purposes. And then it will release what it doesn't use, into the general circulation. Most amino acids are actually digested and absorbed. We don't really see a lot in the feces, or we shouldn't. Let's just say that. In terms of excretion, if we have a buildup of ammonia, which we'll talk about in the future, we become toxic. We get encephalopathy, which means we get confused. And so we need to convert that ammonia to urea to get rid of it for excretion in urine. That concludes the lecture on digestion of proteins. I told you it was fairly straightforward, and I will see you next time. Take care.